origin of life in terms of the RNA world. First, let us see the process of the origin of life. A key event is the emergence of the first Darwinian entities. Then is long, complicated evolution of Luca. Thereafter, certainly, is further evolution toward modern life. Additionally, we know that before the emergence of the first Darwinian entities, there should have also been some kind of chemical evolution. Then, the evolution during the origin of life should comprise both of them. However, we know that the pre-Luca Darwinian evolution should be the major portion. Then, we are curious about what kind of Darwinian entities should have been involved in such early evolution. A major clue is that they should have been much simpler than our modern organism. Indeed, for Darwinian evolution to start, two key features were required, that is, the heredity and the encoded function. Now that RNA can implement both of them, then perhaps initially both proteins and the DNAs were unnecessary. Then came the hypothesis of the RNA world. Then, how simple on Earth could Darwinian entities in this world be? Now, computer modeling work has given us some ideas. First, RNA molecules themselves may have acted as Darwinian entities. That is, a ribosome may function to favor the replication of itself and thereby became thriving in the prebiotic pool. For example, an RNA polymerase ribosome that catalyzes the template-directed copying has long been suggested to be such a candidate. Such a thing has been supported by computer modeling work. However, in logic, the RNA replicates should not be the only candidate. For example, a nucleotide synthetase ribosome may also favor the replication of it itself by synthesizing nucleotide around itself. A modeling work of ours has supported such a thing. In fact, if we emphasize the potential for Darwinian evolution, the random RNA sequence has already been Darwinian entities. No matter how, after the emergence of those functional RNAs, they may cooperate in the naked scene. For example, a modeling studies has demonstrated the cooperation of the RNA polymerase and the nucleotide synthetase in the naked scene. Then is the transition from the molecular to cellular form. For the functional RNAs in the naked scene, actually, there were two key problems. The first is parasites, that is, RNA sequences without adding function, but can exploit the ribosome in their replication, thus competing for raw materials. The second is cooperation. That is, the ribosome should be adjacent enough to conduct cooperation. Spatial limitation is a solution for both the two problems. For example, mineral surface or something alike has been suggested to provide such spatial limitation. However, such spatial limitation is in itself quite limited. Obviously, a definite boundary is a more effective solution. Then came the RNA-based protocell. In fact, the transition from molecular to cellular form has been referred to as the first major transition in the history of evolution. This is our modeling on such a transition. Here, the ribosome, denoted by the blue dots, in the central naked area, becomes thriving. Then, when an empty vesicle took up some ribosomes, perhaps by chance, the RNA-based protocell becomes thriving, and more thriving, and finally, the central naked area became deserted. Then is the emergence of unitary protocells. Initially, in fact, the ribosomes, or say genes within the protocell, replicate separately. Then, when the protocell divides, some genes with less copies may have got lost. Certainly, a solution is the linking of genes, that is, the emergence of a proto-chromosome. The genes were linked and replicated together, but produce the ribosomes separately to conduct functions. The evolution of the gene linkage has also been modeled theoretically. Additionally, one of our modeling studies have suggested that the proto-chromosome should have been a circular one, and they could produce ribosomes separately by self-cleavage, just like the situation in modern viroids. Then a DNA chromosome may have taken place of the RNA chromosome. That is the so-called genetic takeover. One of our modeling studies have suggested that the DNA's feature of stability, template suitability, and high fidelity in replication, all these merits may have played roles in the genetic takeover. But notably, also by modeling studies, it has also been suggested that neighbor division, DNA as genetic material and RNA as functional material, especially per se may have driven the genetic takeover to confront parasites, perhaps as early as in the naked scene. The advent of the proteins was inevitable because of its variety of potential in functioning. But this is a complicated evolution. It should include the origin of a proto-translation system, perhaps include proto-tRNAs and proto-RNAs. Additionally, adding serial aim to explain the origin of translation system should simultaneously explain the advent of genetic coding. For example, both based on the direct RNA template theory for the code's origin, we have proposed a DRT-RP hypothesis on the origin of translation system in the RNA world. Corresponding modeling work is underway. Certainly, the computer modeling on the early evolution can also extend it to the so-called chemical evolution. For example, this is our modeling on the origin of homochirality in early evolution. Here, the two types of nucleotide, L type and D type, were represented by the blue dots and the red dots, respectively. Then, during the chemical evolution, D type, one type of chirality, here is the D type, achieve an advantage. Then, with the advent of a ribosome and with the spread of the ribosome, the chiral deviation was enhanced. That is, finally, the whole system became almost all red and nearly no blue. Certainly, there is also experimental work. Experimental work in this area majorly deal with the chemical mechanism before the emergence of the first Darwinian entities. But some of them has extended to the Peluca Darwinian evolution. This is a recent review on relevant experimental work. Perhaps one of the major flaws in this area is that the computer modeling work and the experiment work has not been brought close enough. Taken together, evolution during the origin of life should majorly refer to the Preluca Darwinian evolution, but it may also comprise the chemical evolution. As it was asserted, evolution is at the center of the origin of life. Based on the idea of the RNA world, majorly via modeling studies, we have gained quite a few insights into this early evolution. And it is expected in future, with the combination of experimental effort and our modeling effort, we would gain much more knowledge on this mysterious but interesting evolution. Thank you very much for your attention.